Hello to all of you. Let's discuss chapter number 10. Let me go to slideshow mode. Okay, so chapter number 10 is about antimicrobial treatment. So basically, the introduction of the modern drugs to control infection, this era actually came in around 1940s. So the new drugs were being developed to control infections. It all started in 1940s. These antimicrobial drugs, they have reduced the incidence of infections, but there are still some infections which have not been eradicated yet. Okay, and also there has been a problem with the development of the resistance to these drugs. So basically, there are many antibiotics available, but there is also the emergence of the superbugs. So the superbugs are bacteria which are resistant to the antibiotics. So there are some bacteria which are resistant to all the antibiotics available. So you cannot treat them with any antibiotic. So basically, the main goal of the antimicrobial or antimicrobial therapy should be that drug should be able to kill infectious agent but not the host cell, and it must be able to easy to administer and able to reach the infectious agent anywhere in the body, be absolutely toxic to the infectious agent and absolutely not toxic to the host, remain active in the body as long as needed, and safely and easily broken down and excreted. These are some of the characteristics of the ideal antimicrobial drug. So the drug is toxic to the microbe, non-toxic to host cells. The drug is microbiocidal rather than microbiostatic, which means it should be able to the bacteria, not just slow its growth. Relatively soluble, functions when highly diluted in body fluids. Remains potent long enough to act and is not broken down or excreted. Does not lead to development of antimicrobial resistance. So this is the main problem with these drugs. Complements or assists the activities of host defenses. Remains active in tissues and body fluids. Readily delivered to the site of infection. Reasonably priced. Does not disrupt the host's health by causing allergies or predisposing the host to allergies. And this is some of the terminology which you should know for the infection of the net risk. Antimicrobial chemotherapy, it's the use of drugs to control infection. It's basically a broad term, so it's all-inclusive for any antimicrobial drug. Antibiotics, these are substances produced by natural metabolic processes of some microorganisms that can inhibit or destroy other microorganisms. Generally, antibiotics term is used for that chemical which kills the bacteria. Semi-synthetic drugs, these are the drugs that are chemically modified in the laboratory after being isolated from natural sources. So they are chemically modified. Synthetic drugs are the ones which are produced entirely by the chemical reactions. Now, narrow spectrum or limited spectrum antimicrobials are the ones which can kill a narrow range of microorganisms, like a drug which is effective against a gram-positive bacteria, or a drug which is effective against a gram-negative bacteria. So those are narrow spectrum antimicrobials, and there are some broad spectrum antimicrobials. They are those antimicrobials that are against wide variety of microbes, like a drug which is effective against gram-positive also and gram-negative also. So there are some antibiotics which are broad spectrum antibiotics, and there are some antibiotics. So some terminology you need to know. So basically, what is the origin of antimicrobial drugs? So antibiotics discovered by the scientist called Alexander Fleming. So basically, he was a microbiologist, and what he found that he had a plate of he had a petri in his dish. He saw there was mold growing in his petri dish. Wherever mold was growing, the bacteria was not. So which means that, so from there he figured out that it's secreting some chemical which is killing the bacteria. So antibiotics are natural metabolic products of bacteria and fungus. They are produced to inhibit or compete. Penicillium was the first mold was mold in the Penicillium and cephalosporium. The importance to be considered before therapy can begin: the identity of the microorganism causing the infection, the degree of microorganism susceptibility to various drugs, and the overall medical condition of the patient. Now, what should be the goal of antimicrobial drugs? So, antimicrobial drugs, they disrupt cell processes or structures of bacteria, fungus, or protozoa, and they also inhibit virus replication, interfere with the function of enzymes required to synthesize or assemble macromolecules, destroy structures already formed in the cell, and they are selective cells without damaging cells. This is very important. That the penicillin is an antibiotic which basically kills the bacteria by inhibiting the synthesis of the cell of the bacteria. Okay, so because the human cells, they do not the cell wall the human cells, they lack the so the bacterial cell wall has a chemical called peptidoglycan. So penicillin basically attacks that. So human cells lack that chemical called peptidoglycan. So that is why human cells are not affected by this drug, by the antibiotic. Drugs most toxic to humans. So the drugs that act upon structure common to both the infectious agent and the host cells, they are toxic to the cells. So there are some drugs. Which can there are some drugs which basically act on like a cytoplasmic membrane. So the cytoplasmic membrane is there in the host cell as well as the infectious agents. So those kind of drugs are toxic to the human cells. Okay. So now let's talk about how do these antimicrobial drugs, how do they basically kill the infectious agents, or how do they kill the microbes. So these antimicrobial drugs, they basically kill the microbes by either inhibiting the cell wall synthesis, or they inhibit the nucleic acid structure and function of the microbes, or they inhibit the protein synthesis in the microbes, interference with cytoplasmic membrane structure and function, inhibition of folic acid synthesis. These are different ways how these antimicrobial drugs, they kill the microbes. So this is the list of some of the drugs, some of which basically target the cell wall synthesis. Examples is like penicillin G and V, ampicillin, amoxicillin, then cephalosporins. This is some of the list of the drugs which can target the cell wall, and this is some of the list of the drugs which can target the which can inhibit the protein synthesis. So they act. So protein synthesis is a process which involves first transcription, then translation. So in the process of translation, there are transfer RNA involved, there are ribosomes involved. So these drugs, they basically go and target different parts of the protein synthesis machinery, and that's the way they inhibit the protein synthesis. So this is some examples are given here. You can go over them, and they talk about exactly which part of the protein synthesis machinery they target, and they inhibit protein synthesis. Drugs that target folic acid synthesis. Sulfonamides, they interfere with folate metabolism by blocking enzymes required for the synthesis of tetrahydrofolate, and tetrahydrofolate is needed by the cells for folic acid synthesis and eventual production of DNA, RNA, and amino acids. And these are some of the example of the drugs. Drugs that target DNA and RNA. Sulfonamides, it interferes with folate metabolism by blocking enzymes required for the synthesis of tetrahydrofolate, and tetrahydrofolate is required, which is needed in the cells for the folic acid synthesis and eventual production of DNA, RNA, and amino acids. Okay, these are some of the drugs that can target cell membrane, polymyxins. They interact with membrane phospholipids. Plasma membrane is made of phospholipids, so these drugs, they interact with the phospholipids, distort the cell's surface, and cause leakage of protein and nitrogen bases. Okay, so the agents to treat the fungal infections. So fungal cells, they are eukaryotic cells. Now they are very close to human cells. So the similarities between fungal and human cells means that drugs toxic to fungi will harm human tissue also. So drugs designed to act on bacteria are ineffective against fungus. So the drugs which are given to the bacterial infections, they are ineffective against the fungus. Okay, this is the list of some of the drugs which are used to treat the fungal infections, and this is their mode of them. Anti-malarial drugs, quinine. So this is the most important drug which is given to treat the malaria, and it has been used for hundreds of years. And quinine is actually replaced by less toxic synthesized quinones like chloroquine or primaquine. This is artemisinin. This is also given for malarial treatment. Now the drugs which are given to treat the protozoal infections. Metronidazole. This is widely used as an amebicide. This is used to treat the infections which are caused by the amoeba. So this drug treats the intestinal infections and hepatic disease caused by Entamoeba histolytica. Also treats giardia and Trichomonas vaginalis. And this is the list of some other anti-protozoal drugs. Challenges of anti-helminthic drug therapy. So flukes, tapeworms, roundworms, these are all large parasites, and their physiology is similar to humans. And let's see what are the drugs used for them. So this is some of the drugs which are used to treat the helminthic. Mebendazole and albendazole inhibit microtubules of worms, eggs, and the larvae. Pyrantel paralyzes the muscles of intestinal worms. Then praziquantel, tapeworm and fluke infections. Ivermectin. These are all drugs which are used to treat the helminthic infections. Now the last is viruses. So the antibiotics which are given to treat the bacteria cannot be used for the viruses, and usually viruses they use the machinery of the host to replicate. So disrupting viral metabolism requires disruption of cellular metabolism of host, because they use mostly the host cell metabolism. They use the host cell machinery. So infectious agent relies on a host cell for the vast majority of metabolic functions. Disrupting viral metabolism requires disruption of the cellular metabolism of host. So basically that is why it's not easy to treat the viruses with the drugs. So usually for the viruses, actually vaccines are used. So all these hepatitis, they are all treated by the vaccines. There are some antiviral drugs, and they inhibit the virus at different stages of their application, or basically they can go and inhibit the viral different stages, like inhibition of virus entry. There are some drugs which can be used to inhibit the entry of the virus into the host cell. Like there are some drugs which are against receptors or fusion uncoating inhibitors. Like these are some of the examples given here. Then inhibition of nucleic acid synthesis. Again, there are some examples So inside the cell, virus basically um, inside the cell, the virus replicates is a synthesis of the nucleic acid of the virus. So that can be inhibited by using all these drugs. And inhibition of viral assembly, own DNA or RNA, whatever the virus is, and then it makes its own capsid, and then the assembly takes place, and then the virus is released from the host cell. So using these drugs, some of these drugs, uh, the viral assembly or release can be inhibited. So I think that's all for this chapter.